Welcome to CRISPR Explained, Gene Editing History, Technology, and Applications. My name is Damon Tai. I'm Bio-Rad's Curriculum and Change Specialist for the Western region of the United States. If you'd like to learn more about our products and services, you can contact us at explorer@biorad.com or follow the link in the description. Throughout this presentation, we will discuss a number of CRISPR applications that can be used as hooks for your students and serve as bridges from your existing curriculum into this exciting technology. We will also discuss the mechanism of CRISPR-Cas9 and demonstrate a hands-on modeling of Cas9 function that can be used in the classroom. We are currently in the age of genome engineering, and technically as a species, we've been there ever since we started messing around with breeding things. Our technologies have grown over time, and CRISPR represents the largest technological jump to date. With its extreme precision, low cost, and ease of use, we start to only be bound by our imagination when it comes to genome engineering. You've probably seen engineering process diagrams similar to these. If you think about the impact CRISPR has on these processes in the context of genome engineering, it affects the entire process, because for the first time, we can imagine editing with high precision, cheaply and quickly, which allows us to plan solutions we previously couldn't have. We're also getting to piggyback on about 4 billion years of engineering in the form of evolution. With this immense potential comes immense responsibility. The stories that dapple this presentation should be thought about as engineering processes first and foremost that happen to use biological tools. You should think about how these stories connect to things you are already doing in the classroom and how they may offer an avenue to enter the subject with students. What do you need to know for the age of genome engineering? Well, a lot of it you're already covering in your classrooms. The basics of DNA and RNA, how those lead to proteins, the structure and function of those proteins, how cells work, and a little physiology, systems biology doesn't hurt either. When I walk into classrooms and give talks on the history and future of biotech, I notice that a lot of students come out of their normal stupor when we hit the de-extinction projects. This and designer babies will likely be your strongest CRISPR application hooks for your students. One of the most notable de-extinction projects in the United States is an attempt to bring back the woolly mammoth, an animal that went extinct about 5,000 years ago and could be helpful in terraforming Arctic regions into grasslands so they don't dump methane into the atmosphere and accelerate global warming. There are two teams working on this project. Beth Shapiro at UC Santa Cruz is interested in bringing back the whole mammoth, where George Church's group at Harvard is interested in transferring 14 genes from the mammoth into the Asiatic elephant to cold adapt it to northern latitudes. The passenger pigeon may be the first de-extinction project we see work in the United States, and that's mainly because we have some really good DNA to start with, as the last one literally died in someone's hands at the Cleveland Zoo on September 1st, 1914. The ecological reason someone would want to bring back this organism is it was a keystone species of eastern forests. Flocks were so big they blocked out the sun, and when they scavenged in the forest, they made major disturbances that many plants evolved with. Bringing back the passenger pigeon will likely have to use the band-tailed pigeon as a surrogate for CRISPR-modified embryos, much in the same way the Asiatic elephant will be used for the mammoth. CRISPR could be used to bring species back, but it could also be used to drive some organisms that humans find a threat to health towards extinction, or at least under strong local control. We have controlled populations of dangerous vectors such as tsetse flies and mosquitoes with a number of techniques that focus on producing infertile or failed offspring. These techniques have been useful but are self-limiting, requiring constant manufacture and deployment of engineered breeding stocks. Gene drives can increase the success by propagating a lethal gene or other such genetic cargo into all of the offspring and not being subject to that roll of the dice seen in normal inheritance. Gene drives work at a molecular level by propagating genetic cargo to multiple sites in a genome. Thus, offspring will be guaranteed to have the cargo no matter which chromosome is passed on. Instead of trying to wipe out the vector, gene drives could also be used to make species that act as reservoirs for disease resistant to such diseases. Borrelia bacteria, the causative agent of tick-borne Lyme disease, is present in at least 43 states and leads to 300,000 new cases a year. A group at MIT is looking to use gene drives to vaccinate white-footed mice. These rodents are the primary reservoir of the bacteria, and small success has been seen with hand-vaccinated mice, but it's not scalable. A gene drive system that allows the propagation of antibodies against Borrelia could allow for a dramatic reduction in the bacteria after just a number of breeding seasons. But what do humans really want to modify? You guessed it, humans. We have been doing selection of offspring for a long time via mate choice and contemporary techniques that allow a pregnant woman to screen for healthy offspring. But maybe you don't want to just screen against a major chromosomal abnormality like trisomy 21. Maybe you want your child not to have the cystic fibrosis mutation you carry. Maybe you wish your child had brown eyes instead of blue. Or maybe you wish your child had a cognitive advantage over others. It's a slippery slope. CRISPR can be performed on somatic cells and germline cells. In the case of editing offspring, you're not only affecting your child, but all of their children as well. Within the past few years, we've already tiptoed into the latest round of eugenics, basically manipulating the gene pool to select for certain kinds of people. When you hear the word eugenics, who do you usually think of? If you're in the United States, it's probably Hitler. But who was doing it right before Hitler? We were. Cold Spring Harbor Laboratory has some great resources on this. Two cases I like to highlight with students are 1927 Buck versus Bell, where the Supreme Court ruled that it's okay to sterilize women that have given birth to 2 or more feeble-minded children, and Skinner versus Oklahoma of 1942, where a judge stops a man from being castrated for his third felony, which was stealing chickens. You have to remember, at this point in time, we thought felony behavior was an inheritable trait, and so to make a better society, castrate the criminals. The topic of modifying people is one of the easiest places for most people to enter the conversation around genome engineering, because they immediately feel skin in the game about what we should be doing with these technologies. So oftentimes, this is a great place to start with students. I often like to step them through a couple of questions, first starting with them as parents. What would they choose? Then have them play the role of the child that was edited. What does that feel like? These first 2 prompts help engage the students emotionally before scaling back to have them think more broadly about what edits a society may want to allow or not allow. Then have them zoom out even further and think about how does this affect the overall gene pool and how does eugenics come into play. It will likely be a muddy but memorable conversation for you and your students. A lot of times, students might get the impression that these technologies just showed up out of nowhere. But remember, we didn't get here overnight. It's important to lay out general histories of transformative technologies for students so that they realize these arise through an evolving process of scientific investigation and technological innovation, very similar to those engineering process diagrams we started with. In 1978, we had the first human born through in vitro fertilization. And this is big because it's removing the eggs and sperm, combining them externally, and implanting them back into the woman, which allows for potential manipulation of those germline cells. In 2012, CRISPR-Cas9 hits the scene, and by 2015, China has already published a paper on modifying non-viable human embryos. The watershed moment, though, is 2018, when Dr. Jingkui He announces that he has modified 3 girls' genomes to make them, hopefully, not susceptible to HIV. And when it comes to the history of DNA editing technologies, it too was a slow progression. In 1968, restriction enzymes are discovered. They are constrained by only recognizing a limited number of locations and being able to cut there. This quickly leads to the first big think tank on genome editing, the 1975 Asilomar Conference on Recombinant DNA, which sets ground rules for what is okay or not okay to clone and other safety precautions, all done with the input of the public. Recombinering technologies come online in the 1980s, making it easier to move specific pieces of DNA into a location where a cut has been made. And shortly thereafter, zinc finger nucleases show up. These are modules of 30 amino acids that interact with nucleotide triplets. By stringing together different zinc finger moieties, one can create zinc finger nucleases that specifically recognize sequences of DNA and can attach there and cut. These are expensive and time-intensive. In the late 2000s, TALENs come online. These transcription activator-like effector nucleases allow for recognition of individual nucleotides, and cloned together in series, it's possible to program TALENs that can recognize chosen sequences and cut there. They are unfortunately a pain to build, and delivery isn't easy. Why then is CRISPR so exciting? It's the low cost, the potential for highly targeted editing, and high ease of use. So much so that you could do CRISPR in your classroom. CRISPR does make genome editing easier, but remember, it's just a tool in the engineering toolbox, and the systems you are modifying are relatively complex. For example, if I wanted to modify the citric acid cycle in a cell to get more fumarate out of the system, I could add in more succinate dehydrogenase or level down the fumarase. Remember, it's not just about converting A to B in biological systems. It's rates, ratios, and all the other pathways that might be stealing the molecule you're trying to do something with. And just to put this in context, let's zoom out from this tiny pathway and get a look at the bigger picture. This is an oversimplified diagram of some of E. coli's major pathways with the citric acid cycle circled. You quickly can see how interconnected the pathways are, and that modifying one position has ramifications potentially at a great distance. So at this point, it might be good to pause the video and think about these questions. What should be our biggest concerns about genome engineering? And how can we prepare students for the decisions we will make as a society with these technologies? A good number of people, when ruminating on these questions, will wrestle with some grim prospects, and that is on purpose. Humans have a strong optimism bias that disables many from thinking slow and calculatively about a phenomenon. And so occasionally peppering in a little, how could this go wrong, helps some people focus. But too much can shut some people down, so let's look at some non-doom and gloom scenarios. One of the most exciting applications of CRISPR is in the field of CAR T cell therapy, where CRISPR can be used to focus the power of the immune system on cancer or another disease. The way it works is T cells are collected from a patient and edited with CRISPR so that the chimeric antigen receptor on the cell can recognize a specific antigen on, say, a cancer cell. If a bunch of these cells are grown up and infused back into the patient, they can focus the immune system at fighting that cancer. Another non-germline CRISPR-based therapy could target patients with sickle cell, and this scenario is part of a capstone activity in Bio-Rad's out-of-the-blue CRISPR kit. Hematopoietic stem cells could be harvested out of a patient's bone marrow, modified using CRISPR to replace the mutation causing sickle cell with the wild-type form. Grow up a titer of these cells and reimplant them, which could skew the patient's ratio of normal-shaped red blood cells to sickle-shaped ones. If you're worried about CRISPR-modified foods ending up in stores around you, you're too late. This pizza has two notable CRISPR-modified foods on it. DuPont in 2012 started producing Cheez-It Swift, which was a cheese culture that used CRISPR to fight phage attack in cheese production. Many people don't like mushrooms that brown after being cut, and so engineers in 2015 used CRISPR to knock out the polyphenol peroxidase that causes browning in cut button mushrooms. Since neither of these are adding things to the genomes of the foods, the FDA doesn't require them to be labeled. Since students are often very interested in what they eat, I like to give them a chance to play around with how they might design crops to better fit their needs using CRISPR. Take one of these crops and come up with a modification that you would like to see. On the next slide, we'll look at a few of them and talk about where they are on their way to market. Apples are, not surprisingly, being modified so that they don't brown when cut. If you could do it in a mushroom, you could do it in an apple, and same thing with potatoes. But they're only at proof of concept at this point. If you've ever seen the chemistry involved in making decaf coffee, you may never want to drink decaf again. So groups have been working on making a decaf coffee without all of the chemical processing. I add broccoli to the list only because so many students hate broccoli, that they often design it out of existence or add a ton of sugar to it, and it's kind of fun to see where their brains go. An important story when it comes to genome editing in agriculture is the attempt to make a hornless cow. Recombinetics, the company who was trying to make the hornless cow, after much work got the phenotype they were striving for and submitted it to the FDA. Unfortunately, the company didn't check their work by genotyping. The FDA found bits of their cloning vector and an antibiotic resistance gene left behind in the genome of the cow and promptly rejected the hornless cow, which was a very, very costly mistake for the company. And that is why any edit with CRISPR should always be confirmed by genotyping. How does CRISPR-Cas9 work? CRISPR, clustered regularly interspaced short palindromic repeats, comes in many flavors in the biological world. At its core, it is a bacterial adaptive immune system. When invaded by viruses who inject their DNA into bacteria, primary immune system components like restriction enzymes can cut up the invader. Some portions of the invader are then captured and added to the clustered regularly interspaced short palindromic repeats in the genome as a way to save a memory of what that invader looked like. The bacterial cell and all of its offspring now can produce a guide RNA molecule that matches the sequence of the previous invader. This guide RNA couples with a nuclease known as Cas9, and if that same virus attempts to infect the cell, the guide RNA can recognize it and Cas9 can quickly cut it, destroying the virus's ability to replicate. Biotechnologists quickly saw the power in the system because if you could program just a 20 nucleotide sequence as part of that guide RNA, you could send a restriction enzyme to any location you liked within the genome. And if you can cut, you can add to or delete from the genome. The structure of CRISPR-Cas9 system is relatively simple and demonstrated in this diagram. Cas enzymes are a class of cutting enzymes involved with CRISPR. Cas9 is the one you will most frequently hear about in relation to CRISPR technology right now. The single guide RNA is a chunk of RNA with structure that allows it to bind with Cas9 and contains a 20 base pair sequence that gives the system its high specificity by binding to a complementary 20 base pair sequence in the target DNA. The PAM, or protospacer adjacent motif, is a 3 nucleotide sequence that is just upstream of the 20 base pair target DNA. The protospacer can be any base followed by GG. In order to visualize how the CRISPR-Cas9 system works, the Out of the Blue CRISPR kit has a hands-on modeling activity. You'll be working with 3 pieces: a strip of DNA, the single guide RNA with its 20 base pair target sequence, the Cas9 enzyme including its guide RNA binding area, a PAM sequence recognizer, and nuclease activity symbolized by scissors. The first thing that takes place is the single guide RNA binds to Cas9. Next, the Cas9 and single guide RNA complex loads a strand of DNA and begins probing it for the 20 base pair sequence and a PAM sequence highlighted in green. Note that the PAM sequence is 3 bases long, and some PAM sequences may use the same base of a PAM sequence right next to them. The DNA strand moves through the complex until the 20 base pair sequence matches a complementary sequence in the target DNA and the PAM sequence matches. At this point, the nuclease engages and causes a double-stranded break. CRISPR provides the cut, but other techniques are needed to complete the editing. Organisms do not like to have double-stranded breaks in their genomes and will work quickly to repair them. A double-stranded break in a bacterial genome for too long is usually lethal. Homology-directed repair, often called HDR, can take place if that cell finds fragments of DNA that look similar to the area around the break, and it will stitch copies of that into place to resolve the break. This is by far the most useful repair mechanism because it gives the biotechnologist the power to add things to the genome. The other repair mechanism is known as non-homologous end-joining, and is a sloppy stitch together of the double-stranded break that can result in random small deletions or insertions. In common speak, people will often refer to this entire process of cutting and repairing as CRISPR. After students have had the chance to do the paper model of how CRISPR works, it's worthwhile to dig into the mathematical modeling how accurate CRISPR is. The Out of the Blue student guide can scaffold them through this exploration and is really important because students get to refresh some math skills and use it to learn how mathematical modeling allows us to predict some of the complex behaviors seen with CRISPR. If you are interested in editing the human genome, which is about 3,234,830,000 bases long, and wanted to figure out what the chances are of seeing sequences of various sizes, I would start by using the table provided. If we are interested in just seeing how frequently a single nucleotide showed up randomly in the genome, we first need to calculate its probability. Since there are 4 different nucleotides, A, C, T, and G, the probability of seeing A is 1 in 4. If we then multiply the human genome by this probability calculation, we are given how frequently, at random, this sequence should occur, which is 808,707,500 times. If I was then interested in the sequence AC, I would multiply that 1/4 probability by another 1/4 probability. I can thus see that raising 1/4 to the power of the number of bases in my sequence and multiplying that by the size of the human genome tells me how many times I can expect to see this sequence in a perfectly random genome. Since CRISPR uses a 20 base pair sequence to target the genome, we can run our calculation, the 1/4 probability raised to the power of 20, and what we see quickly is that at 20 bases, the probability of finding that sequence in a genome the size of a human is less than 1, which means this is really accurate in a perfectly random genome. However, genomes aren't perfectly random, so we have to take our results with a grain of salt. Off-target cutting is one of the biggest concerns with CRISPR. How might this happen, and how might it be stopped? These are good questions to have students think over after the mathematical modeling activity and to discuss. Off-target cutting could take place because you may be cutting a sequence that occurs more than once in the genome due to the non-random construction of the genome. More complex mathematical models that are based on the sequence of the human genome could provide better insight to designing guide RNAs that land in only one location. One of CRISPR's most important uses, though, will be one that doesn't get covered much by most sources, as it isn't very sexy, but it is the cornerstone of how we understand organisms in the molecular era. That is using CRISPR to disrupt genes so that we can understand their function. A stellar example of this was done recently at UC Berkeley, where they disrupted developmental genes in butterflies, which drastically caused changes in their wing patterns. Previously, this sort of work would have taken years, but CRISPR made it easy to happen in a very short period of time. This is really important for the human genome because we still don't know what a vast majority of our genes do. With the Out of the Blue CRISPR kit from Bio-Rad, you and your students will be able to use CRISPR in the classroom to disrupt a gene that can be monitored with a color change from blue to white. How do you know for sure that you disrupted the gene in the way you intended it? We don't want any hornless cow situations. Genotype it with Bio-Rad's Out of the Blue Genotyping Extension Kit. Now that we've taken care of the hard part, all you have to do is think about where does CRISPR-Cas9 gene editing fit into your curriculum.